Dum dum dum. All right, let's see where we are at. I have my intrepid genetics assistant. Isn't she adorable? All right, sorry. I'm a bit of a crazy morning today. So I started the room a little late, so just give a few people time to join, sort themselves out. I'm going to find my board eraser. My cat didn't run off with it. Here we go. Okay. All right, peeps. So the plan for today is to finish off PowerPoint 11, which is like the nuggety details of DNA replication. And then it's pretty much open session for as long as people want, because that's pretty much it until, well, the exam will be next Tuesday, so we'll have a lecture for the new section, like the third section, but that won't be on the exam. So today is the last material and the last chance to talk about the material for exam 2. Okay, so I'm going to bring up this. Come on, down you get. There we go. All right, so first up, we finished up just here. Come on, there we go. Does anybody have any kind of pressing questions or stuff that they'd like to go over or clarify for what we covered on Tuesday? Oh, that's good coffee. Nope. Crickets? I wonder where Angela is. She's normally... Oh. All right. Anyway, so no questions so far? Okay, well, if any do occur to you, please do pop up and ask them, either in chat or in, just unmute your microphone and shout them out. I'm totally fine. Okay, so this is where we ended up at on Tuesday, which is going over the kind of the differences between prokaryotic and eukaryotic DNA replication, right? And so the principles remain the same, but there are some key differences. Chromatin structure is one, DNA polymerases and how they work is another one. Particularly this one here, DNA polymerase alpha, which is both, there's Angela. I wonder where she was at. DNA polymerase alpha, which is both primase and the first DNA polymerase, and then it hands off to either delta or epsilon, depending on which strand is being synthesized. Another difference between prokaryotes and eukaryotes is, oh yeah, no worries. We're just recapping over the last little bit we did on Tuesday. And then the removal of the primers and filling in the gaps is also different. So in prokaryotes, it's DNase, shit, I always forget, DNase 1? Yeah, there we go. Not DNase, DNA polymerase 1. That removes the primer and fills in the gap. In eukaryotes, the primer is kicked out by RPA, which is replication protein A, I think, off from memory. And then the existing DNA polymerase kind of continues on until that gap is filled. Now, here's a really cool one, right? So this is kind of one of the reasons why you get old. Obviously, not dying young is, the big reason why you get old, right? Living fast and all that sort of stuff. But one of the reasons why you appear older as you get older, right? You just don't stay looking the same. Particularly later on in life, around the 50s, 60s, is what's called senescence, right? So it's basically cellular retirement in a nutshell. It's actually quite considerably more complex than that, and I don't pretend to truly understand it. But essentially what it means is that those cells have hit their replication limit, which is called a Hayflick limit. Crap, I can't actually see what I'm writing. Oh, no typos. See if I can spell this right. And roughly speaking, it's not a hard number, but roughly speaking, it's about 60 rounds of cell division. So some of your cells are perpetually replicating, and we'll talk about how they do that in a second. Others replicate up to that limit, and then that's it, they're done. And so I think muscle cells actually are kind of included in that. So after a while, you are unable to keep replacing damaged muscle cells, for example, when you get old enough, once they've done it enough times. And again, I'm kind of waving my hands around here because aging is a massively complicated subject. And then you also have cells which are able to, you have perpetually dividing cells, so those are stem cells essentially. Normal cells which can replicate a set number of times, or divide rather. And then you have other cells which essentially, once they've differentiated, they're done. They're called post-mitotic cells, and your neurons are a good example of that. So once you have all of your neurons as a wee baby, that's it. You don't get no more. Actually, you lose quite a considerable number. I think like about 25, 30% before reaching adulthood. It's called pruning. It's like, yeah, I don't need these brain cells. Boop, off they go, dead. Anyway, and that's also why brain injury is such a hard thing to recover from, because you can't grow new brain cells to fill in the gaps. If once they die, they're dead, they're gone. And so most kind of post-brain trauma recovery is actually more about utilizing other parts of the brain to do the same job as the missing or dead parts do, rather than actually, kind of like bone healing type recovery, right, where you fix the damage. So that's kind of interesting. Anyway, so I've gone off on a little bit of a tangent. So the issue with linear chromosomes is what's called the end replication problem. And that's a really nice image here, which helps explain that. And so basically, the end replication problem is caused by primers at the end. So if you look at this kind of image at the top, right, we've got 3 different replication bubbles that eventually merge. We have all these different fragments which get filled in. But how there's no DNA polymerase here, right, to the right of the top strand to fill in the gap or to remove the primer and then fill it in. Similarly, over here on the bottom left-hand side of the bottom strand, same problem. So there's no way to fill in that little bit of DNA, right? Okay, so kind of hold that thought in your head. So what that means is that that RNA has to be removed. I'm not entirely sure how, probably by some RNases, I guess. But the result of that is that each round of cell division, one end of the newly synthesized DNA strands, right, for both chromosomes, will be shorter by the length of the primer. And you may think, okay, so what? Primer's like 20, 25 base pairs long. But if you think about it, if you continue that for long enough, oh, hang on. Let me explain the last little bit that's important. So the issue then becomes, right, so this is kind of the beginning of the problem, right? Now you need to think about what happens the next round of cell division. So this strand, the original parent strand, the template strand, gets replicated as before. What about this one? This one will also serve as a template, right? Because you split those 2 strands apart and each one serves as a template, semi-conservative replication. So this one will also serve as a template, but it is shorter. And so the new copied strand will start from this end, not the original end of the original parent, but the new end of the new parent strand. Does that make sense? Yeah, again, there in a second, Angela. So essentially, every time your DNA replicates, it gets shorter, right? And so if you actually look at a young person or even a baby's chromosomes, they will be longer than an adult. And a middle-aged adult would be their chromosomes will be longer than someone who's 80 or 90, for example. Because every time their cells divide, the chromosomes get shorter. And so there are a couple of different ways of getting around this. One, as Angela just pointed out, is to essentially make the ends of those chromosomes like buffer zones, kind of like those water-filled barrels or whatever they put in front of bridges and highway dividers and stuff like that. Right, so they basically don't do anything. The telomeres have no coding DNA in them. They don't. None of the DNA in telomeres code for proteins, and actually, it's highly repetitive. It's just kind of just the same sequence over and over and over and over again. And so it doesn't matter that your chromosomes shorten, at least initially, because you start using up that buffer region, right? The reason for that limit, the Hayflick limit, is that if you that buffer region is only so large. I think they're usually in the few thousand base pairs, give or take. So if DNA replication continues around and around the cell division, eventually that telomere will be gone and you'll start eating into the actual functional chromosome, right? Into essentially coding genes, which are at the very end of the chromosomes. So before that happens, the cell basically goes, all right, I'm done. Gonna cash in my retirement fund and play golf in Florida or something like that. And that's it. Or whatever retired people do. I think they just go to Florida, right? Anyway, seems that's what it appears like every time I've been to Florida. And like the median age is like 80. Um, anyway, so that's, you don't want to go to Florida. Have you been to Florida? Ugh. Anyway, um, yeah, you could break the mold and go somewhere interesting. Um, so telomeres and the okay, sure, Peyton, no worries. So telomeres are one solution to that problem. Essentially, a buffer zone that has a kind of a trip, like I don't know what it would be, a sensor that says, okay, we've shortened the telomeres too much, and we've got to stop. And so essentially, that cell is prevented from dividing again, ever, right? It is properly post-mitotic. So that's one solution. But probably you're thinking, okay, how does that work for stem cells? Anybody got any ideas? Just put. I want to have a sip of my coffee. Ah, because stem cells divide over and over and over and over again indefinitely. They are essentially immortal, give or take. So how do stem cells deal with this whole telomere shortening problem? No, they don't have shorter telomeres. Actually, their telomeres are beautifully long and robust and shiny. So what they have is hang on a second. I'm not just going to show you the mechanism because it's kind of tricky and I really don't think I do a good job of drawing it out. But you can look it up if you're interested. Oops. Peyton, is your audio any better? No, hopefully it is. Anyway, so essentially what telomeres do, stem cells, sorry, is they have an enzyme which, through some fairly complicated jiggery pokery involving RNA primers and loops and stuff like that, they fill in that gap. And so in stem cells, if you look at if you look at stem cells in, I don't know, like skin stem cells or gut stem cells, whatever, right? In an 80-year-old individual, the chromosomes will be exactly the same length as in an infant. And essentially, they put back those missing gaps. Okay, so now I'm going to kind of get you thinking on a different slant. What other types of cells are essentially immortal that hopefully none of you have, or me, or anybody you might know? Can anybody think? Well, no, not embryonic. I hopefully don't have any embryonic stem cells left. That would be terrible. Cancer cells, right? So, tumor. I'm going to spell it with a U just to annoy all the Americans on here, which is like everybody. Basically, tumor cells have an activated telomerase. Oops. And essentially that's one of the markers of oh yeah. Yeah, I mean that's not to be taken lightly. Actually, the reason why I was away a little while ago is because my wife's uncle died from cancer just recently, which had been fighting for, thank you, for about 6 months or so. I mean, he was 80. He had a very, very good life, which was great. But uh, essentially, those cells never stop dividing, right? Which they should, if you think about it. Because even if you have um, a signal which tells your cells to start dividing again, even though they shouldn't, right? Because most of, like your lung cells, right, you don't tend to need more lung, right? Once you've got enough lung. Um, and even if those cells which start dividing escape being killed, right, which is the kind of the immediate response to uncontrolled cell division in our bodies, right? So that's another marker. So there's like essentially there's three different markers for tumor cells. Even if they get past those two roadblocks, which are pretty robust, right, which is why you tend typically to see cancers start emerging more as you get older, by and large, because those blocks break down. Um, even if those cells get past those two roadblocks to cell division, they would hit that Hayflick limit and stop, right? So I don't actually know how. I'm not sure if it is known, but I certainly don't know it. Somehow telomerase gets activated in those cells, even though they shouldn't be having it on. It's just a stem cell deal. And they're able to repair their telomeres every round of cell division, and so they can divide indefinitely, essentially. And so that's how you can end up with, these enormous tumors, which are like the same size as you are, essentially, right? It should not be able to happen, right? But essentially, this uh, gap fixing mechanism, which allows stem cells to divide indefinitely, gets hijacked, and that's what allows tumor cells to do so. Um, Hang on, give me just a second, Andrew, to read that. Oh, and Alyssa too. So, I'm not 100% sure, Alyssa. So the question was, does this apply to non-cancerous tumor cells? And so I'm not an expert on cancer by any stretch. As far as I understand it, those kind of, what are called benign tumors are kind of like limited run resumptions of cell division. So something turns those cells on again, even though they're in stationary phase, essentially, right? G0. And for whatever reason, those cells don't continue the process. And that could be a lot. It could be because telomerase isn't activated, right? And so warts are a classic example. I've got a honking one on my elbow, which people always talk about for some reason. But essentially, they grow and form like an outcrop of, I don't know, maybe somewhat undifferentiated cells, right, which are not supposed to be there. But they don't continue. It could be because they've hit the replication limit. It could be because another big thing for tumors is what's called angiogenesis. And so essentially you can get, I don't know, I think it's about, where's my webcam? Kind of clump of cells about that big, give or take. And after which the cells at the center of that clump don't get enough oxygen and can't get rid of their carbon dioxide and that kind of deal. So they go anoxic. Give me just a second, Alyssa. I'm still on my train of thought. And so if you don't promote new blood vessel formation, those tumors can never get bigger than kind of that sort of size, give or take, right? And so that's another reason for things like that not proceeding to like the scary, oh crap, I need to have surgery and chemotherapy kind of stage. as far as I understand it, that's my kind of best guess. But certainly, angiogenesis is a really important part of tumorigenesis, right? Because once they get to a certain size, the cells in the middle of that clump will die, right? Because they don't have enough oxygen. And that actually promotes formation of new blood vessels. So that would be my guess. And it is just a guess, but I think it's a kind of reasonably well educated one. Sorry, I'm just listening now. I think I've got a cat that wants to come into the room. So Andrew is asking about the progression of tumorigenesis. So yes, the first step is getting past those tumor suppressor genes, which are essentially cell cycle checkpoint control genes. That's one of the main, categories of tumor suppressors. So essentially, kind of trying to think of the best way to answer this question. So we do have immortal cell lines in the lab. Most of those are accidental. So I don't think that, I'm not sure, because again, cancer genetics is just like this massive field. I'm not sure if we've been able to generate our own immortal cell lines, right? Which is essentially kind of what you're talking about to allow these cells to continue growing. I think most of them are essentially extracts from patient tumors, and HeLa cells are actually the most famous versions of that. You probably have heard about this. There's a lot of controversy about HeLa cells. Oh, is that Jasmine that was asking to come in? I thought so. Let's see if you can see one of my other cats. She's a very can you just see her by the door? She's a very flighty cat. She generally will not hang around. Okay, where am I at? So the very first immortal cell line was extracted from an African American woman who had, I think, ovarian cancer, I think from the top of my memory. And those essentially have been used for the last 70 years, I think. And there are an awful lot of other ones too. So in theory, we could replicate the same processes that allow tumors to form, but it's actually extraordinarily difficult. We have got to the point where we can make little organoids from undifferentiated cells or like pluripotent stem cells, where we can make like little guts and what are the other ones that have been done? Little lung tissues and things like that. But it's not a trivial process by any stretch. Okay, what else? Yeah, well, that's what people were hoping for, to find a way of um, reversing aging, right? But essentially, the reversal of aging is cancer, right? So it's very, very hard. It's not, and really cancer is resumption of development, right? So it's kind of those cells have got an inappropriate signal to start developing again, but they don't have all the other signals around them to tell them what they should be. And so you are kind of the way you are, with like fingers and like hair and all the other bits and pieces that make us functional organisms, because of all these different signals which were present during your development, as an embryo. And so it's very, very hard to replicate those signals and that context after you've already become an adult and stopped developing. So even though it sounds kind of really cool to be able to, well, we could just turn this on and turn that on. Essentially, the outcome of doing so would be cancer, because that's essentially what cancer is. So being able to turn that stuff on and get it to differentiate in the correct way without kind of running off in a direction that we don't intend is a non-trivial matter. And yeah, so even though some of parts of our body, like our skin, hair, our gut, particularly our intestines, continually kind of replenish themselves, there's an awful lot of us that doesn't. And it's the bits that doesn't that cause the problems. Jasmine, what? Ah, damn it, those bloody cats. Anyway, gotta go. I want to give you a little close up of Jasmine because she's the psycho princess. Let's see if she'll hopefully. Oh, my bed's unmade. Never mind. What do you want? Jasmine. Go on then. Get out. Go. Shush. Come on. Anyway, that was the second. Well, that was actually the buy one get one free cat, or the other one rather, which I'm sure wasn't actually a real thing at the Humane Society, but it was good enough for my kids to go, oh, we gotta get them, we gotta get them. So this is the other one. That's Noelle. They were dumpster sisters, basically. They were both dumped as kittens in a dumpster. Oh, she's adorable. Come here. Isn't she? She doesn't like being held, though. Likes being stroked. Hey, you. She's a very, very lovely cat. The other one is a complete nutso. And they're not technically sisters, but they're about the same age, give or take. And this one was the protector. So she protects Jasmine from being beaten up by our other cat. Anyway, it's complex. All right. So anyway, yeah, that was a little bit of a tangent, but it's a really interesting one because, cancer as a general principle. I mean, the details I don't find super interesting, as a scientist at least, because I've got other interests. But the actual process of how it kind of occurs and all the different steps that have to happen are quite interesting. And so this telomere telomerase kind of problem is, another interesting insight into that. Okay, anybody have more questions? Actually, it's funny going to Angela's comment about the fountain of youth. A while back, everybody thought that was a compound called resveratrol. Let's see if I can spell that. I think it's spelled that way, which is basically one of the compounds found in red wine. Supposedly, why people in the Mediterranean live longer than people in the UK. And actually, I think the main reason is because they eat healthily and don't eat a ton of crap like Americans. Sorry, not Americans. British people do. I don't think it's down to red wine at all, but it doesn't stop you from having a drink every now and then. So yeah, that was, and I'm not sure about billions, but certainly hundreds of millions of dollars was poured into research and companies trying to figure out how to use that to make people live longer. And it's essentially died a quiet death. So I guess they didn't find out. Okay, so last thing really at all, right, in this section, even though we have, what do we have? Yeah, we're going to start eukaryote genome structure and organization on Tuesday, but that will not be in exam 2, right? So only what we're studying up till the end of lectures today will be in exam 2. So don't worry about the next PowerPoint after this. I think is number 12. That will not be on the exam. Okay, so essentially what I want to do, and this is I'm not sure how to teach this because it's a very visual kind of problem, but I want to talk about recombination for a bit. And so homologous recombination, HomRecom, essentially does two things. And I'm not going to write homologous chromosomes out in full because that would just take too long and have too many spelling mistakes. Ah, crap. Oh, and again, if anybody does want, I know people sometimes like asking me questions privately or commenting on stuff privately. If you would not like those to be made public, which is totally fine by me because they are private, let me know and I'll remove them from the chat text thing which I post up to Blackboard. Right, I'm not going to do it as a matter of course because it's a lot of work to go in and take those all out, but as a matter of course. But if there's anything that's in there that you don't want to appear, just let me know and I'll take them out. Totally fine with that. Where was that? And phase 1 and allows the repair of DSBs in the lingo, double-stranded breaks, are what one would put if I can spell it right, are catastrophic if they're not fixed. So it's a death sentence, essentially. If a cell has a double-stranded break which is not fixed, then it is told to kill itself. It dies through apoptosis. And the reason for that is even if that doesn't happen to be in a coding region, which it most likely isn't, given how kind of small the fraction of DNA which codes for proteins is, next time that cell tries to divide, it will generate 2 chromosomes, right, from that break, right, because each single little bit will then get copied separately to make a chromosome, and that is really, really bad news. I mean, it does happen, like occasionally in evolution. Sometimes chromosomes get stuck together. I think chimpanzees have 2 of, our last common ancestor, right, led to chimpanzees and to us. And in the chimpanzee kind of branch, 2 of those chromosomes got stuck together to make one larger chromosome. Sometimes they get split, but it's usually a really bad deal. So they can be repaired. HomRecom. And this is a long one. Non-homologous end joining. oops, NHEJ. Right, so I'm not going to talk about non-homologous end joining much because it's not in the PowerPoint. That's essentially the Hail Mary option if this damage is not detected during DNA replication, essentially, right? And that's kind of like if you give someone a bunch of different pieces of string and tell them to stick them together to make a long rope. They'll do so, but you're not going to be guaranteed that that rope is going to look the same as it did originally before you cut it up. So that's the downside of non-homologous end joining. Whereas homologous recombination is very faithful and very accurate. Actually, I wonder, give me just a second. Tum. You might be seeing my tum. Where the hell are my lectures? Oh, here we go. Sorry. I think this is the one that I want. So I'm looking for a really cool here we go. Yeah, that's fine. Okay. Hang on, why is this not working? Give me just a second. This is turning into a little bit more of a That's a good question, actually. Which chapters are on exam 2? Hang on, let me get my schedule back up. So essentially, exam 2 is non-Mendelian inheritance patterns. So this is kind of sex linkage and let me just, actually I can share this with you. Here we go. So yeah, so I don't actually know, to be honest, Angela. It's a long time since I've looked at that book. So essentially, any extensions from Mendelian inheritance, so differences in dominance patterns, so codominance, incomplete dominance, sex linkage, sex-linked inheritance, also mechanisms of sex determination as well. So that's all part of that non-Mendelian inheritance patterns. That is chapter PowerPoint 8 and onwards. Let's see. Just double checking, make sure I don't lead you astray. Oh, sorry. PowerPoint 7 and onwards. Oops, come on. And then linkage, which is a big one. We spent a bunch of time on that. DNA structure and essentially DNA replication in prokaryotes, in eukaryotes, and then homologous recombination, which we're just doing now. Did that help? Cool. All right, let's stop sharing this. And yeah, sorry for the tangent. I was trying to do something which turns out I didn't know how to do. Anyway, so just as a quick aside, this is always funny. I don't know if anyone actually remembers Conan the Barbarian. I watched it many, many, many years ago. But yeah, Arnie, when I don't know, probably better not make any comments about Arnie. But anyway, essentially, Conan the Barbarian, no matter how tough he is, will die from exposure to about 5 grays of radiation, which, come on, how much is about 5 grays? Oh, that's not in grays. That's in a different one. Essentially, 5 grays will kill you within 2 weeks, right? So a large degree of exposure to gamma radiation, typically, like a Fukushima plant or Chernobyl, things like that. If you're part of the cleanup, then most likely you would receive a large dose. But anyway, these bacteria, so these are called Deinococcus radiodurans, which is super neat. They can withstand 5,000 grays quite happily and grow. And the way that they do that is, they have as many as 4 to 10 copies of every gene in separate chromosomes. And so it is able to basically do homologous recombination all the time, whether or not it is dividing or not. And actually, because it's a bacterium, it doesn't have to worry about, prophase and the like. It just gets on with it. So, she was just cleaning herself, weren't she? Okay, so anyway, so essentially, homologous recombination is pretty cool because of both its role in cell division and its ability to allow double-stranded breaks to be fixed and fixed properly too, which is a much more important deal. Because if you can fix them, but if you end up sticking the wrong end of the chromosome to the wrong end, then it might not be much better. And so homologous recombination is really it's a very complex process, right? Not super well understood for a bunch of different reasons. And some of the most important parts is that if you're going to be doing homologous recombination, you want to make sure that each homologous chromosome is precisely lined up, right? You can imagine if it's like so, then essentially if you have a crossover kind of around like my first knuckle here, you're going to end up with one chromosome a lot longer because it's going to get all of this, and one chromosome a lot shorter because it's going to get that little bit, right? And so that is not what you want, and right, so that's what the synaptonemal complex does, at least in prophase 1. I'm not sure about any other time, but that helps line up somehow, again, I don't know how, every single base in those 2 chromosomes. Then you have to break at least one of these chromosomes. I'm not entirely sure if you have to break do a double-stranded break on one and combine it. That's typically how it's been explained, but potentially you could do a single-stranded break on both, maybe. And then you have to fix those breaks by swapping over the DNA, right? So it's quite complex, and it's a tricky kind of process to visualize. So I'm going to go through it here, and then I'm going to try and draw it out, right? See if that will help. Okay, so essentially what happens, let's go start at the beginning rather than middle way through, is you line up those chromosomes, and then we'll just use it on double-stranded break on one, because I think that's probably the simplest way to explain it. And so what you're going to have is one of these chromosomes is going to act as the template, right? Or scaffold actually is a better word. So this chromosome is going to allow the broken ends of this one to be lined up and fixed properly, right? So essentially this is like a kind of if you imagine having like a long sentence written out and you cut it in the middle, right? And you've got no way of really knowing how it sticks back together. If you have another copy of that sentence, then you can kind of line them up essentially to make sure that you have the right end going to the right end and not the other way around, for example. Right, so that the red chromosome in this case is going to act as that scaffold to allow the break in the other one to be fixed. So the first step is to get rid of some of the DNA on that broken chromosome. And this is to make it single-stranded. It has to be single-stranded because it's going to pair with that scaffold chromosome, the unbroken, undamaged one on the other side. And the only way it can do that is if it's single-stranded. Right? If it's double-stranded, there's not going to be any pairing going on. It has to be single-stranded because essentially this strand is going to kind of force its way in to the template strand, like the scaffolding chromosome. So first you make some of this single-stranded on each side. And the second thing is we have what's called strand invasion, right? And this is essentially this single-stranded bit of DNA on the broken chromosome kind of forces its way in to the other double-stranded bit of DNA and base pairs precisely, right? That's why they're lined up so well with its complementary strand in the other chromosome, right? Which essentially kind of leaves this strand flapping around in the breeze, temporarily at least. Does that make sense? It's hard to do with fingers, but I'll draw it out in a little bit. Okay, so essentially once that strand is base paired there, we now have a 3' OH. DNA replication can begin, right? Because we have a template strand and we have a bit of DNA which has a free 3' end. Trying to make sure I see what I'm doing. And so now we can continue DNA replication in this direction, right? Using the original unbroken scaffold or template chromosome as a template to do so. Okay, so that's for one strand, right? So essentially we've got our two strands of our repair unbroken chromosome. We've got our single-stranded bit of our broken chromosome, right? It's single-stranded because, we made it so. And that, kind of forces its way in here and base pairs with its complementary strand on the other chromosome, the red strand, and then starts synthesis in that direction, right? So that's for one strand of the broken chromosome. We have the strand go in the other direction too. And so what this is fixed by is that, I was talking about this one kind of hanging in the breeze, right? Now this is single-stranded. It can now bend, oops, I can't do it with my hands because I can't bend my hands around that far. That free strand of the original unbroken chromosome can now base pair that way. There you go, that's not too bad. With the other broken end of the chromosome. And that's what you see in step 7 here, right? So that kind of bubble that's like hanging down is now able to base pair, or, with full complementarity, right, with the other broken end of the chromosome. And DNA replication starts and continues using that free 3' end. How about that? It's kind of tricky to get. Took me a little while, to be honest. And so what happens is that the one on the bottom continues until it hits the existing strand. And then the one on the top, do I have a good link to a video? I don't, but I can find one though. Actually, we can do that in the break between this and the Q&A session that we'll have after this before lab. I'll have a good look. But I'll draw it out as well in a second, which may help or not, depending on how good my drawing is. But yeah, I'll remember to have a look for that. Yeah, I'll make myself a note. This actually works pretty well for me. I've got a terrible memory. So what you end up having is that both of those broken ends end up rejoining further along on either side the original broken chromosome, which is what you can see in 8 at the bottom. Hey, God damn it, bloody cats. Quit it. Honestly, you probably didn't expect to learn so much about cats, right, when you signed up to take genetics. But the problem now is that those two strands are together, right? They're part of each other. And so we need to separate those before those two homologous chromosomes can come apart and go their separate ways. And those places where they're stuck together are called holiday junctions. And that's not like Holiday Inn. That's holiday as in, I don't know, whoever discovered those junctions. Actually, was the guy who started Gregarious Games in Ready Player One? I think he was called Holiday. I'll have to check that later. Anyway, and so those holiday junctions have to be resolved, right? They have to be broken, which is what we're going to look at here, if it lets me, and then stuck back together, right? So at the top, you see this holiday junction. And also on this diagram, you see we've got different letters, right? And so essentially what you're looking at is a pair of homologous chromosomes with 2 genes in cis, right? So we have both dominant alleles on one chromosome at the top and both recessive alleles on one chromosome at the bottom. And so depending on how those holiday junctions are resolved, using holiday junction resolving enzymes, unsurprisingly, determines where the site of the crossover is. But it will still result in the same recombinant chromosomes, essentially, right? You will still have a dominant and recessive allele on the top and a recessive and dominant allele on the bottom. Whereas before, it was dominant, dominant, recessive, recessive, right? All that matters in terms of how these junctions are resolved is where that crossover point is exactly. The actual genetics in terms of genotypes is the same. What are you doing behind there? Just hiding behind my whiteboard for some reason. Okay, let's see if I can draw this out. Now we have a little helper, I guess. Okay, stick this as far back as I can. Okay, so I'm going to stop sharing this to make the image bigger. Okay, so we have this, and let's do like so. Hopefully you can see this well enough. And I'll probably need to go through at least a couple of panels because I'm not going to fit everything on here. Okay, so essentially this is what we're starting with. Sorry about the reflection. Actually, I'm going to just go turn off that light. Might make it a little darker, but hopefully it will make it a little easier to see without reflection. Okay. There you go, that's a little better. Not quite as bright, unfortunately, but hopefully a little easier to see. All right, so essentially what we're going to do is we're going to break this chromosome here, like so. Right, and so this is a double-stranded break. It breaks both strands. And we're going to be using this chromosome up here as a scaffold to fix this this break. Right, and so first what we're going to do is, actually it doesn't matter which way around we do it. We're going to basically chew back some of that DNA, and we were going to leave it so that the long ends are the 3' ends of that DNA. The reason for that is that we want to extend these ends, right? So that we can kind of fill in, kind of join in and fill in those gaps, right? So we have to have 3' ends because DNA replication or synthesis only occurs from the 3' end onwards, right? Because of that 3' OH. Now, essentially, what happens next, arrow, is, let's stick this like so. This is a lot easier to do on a big whiteboard. What happens now is we've got this, let's kind of draw this up like so. I haven't drawn the other strand up here because I want to leave myself some space. I'll draw it in a second. Oops, made a single strand of break there. So now this strand here it base pairs, right? So there is perfect complementarity or almost perfect complementarity between this strand of this chromosome and the matching strand essentially of the other chromosome. So they are able to go together like so. But in the process, it forces this the other strand out, like so, right? Because you can't have a triplex of DNA. You can only have two strands. And so if this broken strand coming in and base pairing with this strand at the top forces this other strand, the original complementary strand for that chromosome, off. Kind of creates a little bubble. Does that make sense? That's the strand invasion part. Okay, so what now happens is we get a little bit of draw in extension. Oops. So that, oops, crap sticks. I keep holding on to the wrong bits of the board. That invaded strand now is synthesized again in the 3' direction because that's how DNA replication works. And in the process, let me just fill in these gaps, which I did by accident. This displaced strand, right, gets displaced further, like so. You can see what's happening here, right? And so now it is large enough, that displaced part, to do the same, base pairing with the other broken end of the chromosome. Does that make sense? And that is also a 3' end. So we can start adding DNA to this end. Anybody got any questions about this? I need to take a picture of it and rub it off. No? Okay. This might not be help completely, but the hope is that it gives you a little bit more insight to what's going on. Let's take a picture. Okay, so let's start with where we ended off. Okay, so let's make sure put those on the end like so. Okay, so this is where we ended off, right? We've had strand invasion and extension, displacing the unbroken strand here, the red one, and that now is sufficiently out of place that it can base pair with the other broken end of the broken chromosome. And so now we just continue that process. Actually, I'm going to do it on here because I need some space, right? So this is going to continue along like so. And now this end is going to get joined back to that end. Now you can see how we get those holiday junctions, right? So the next step would be to finish that job off and like just stick them back together. Like so. Right, and don't forget we have our alleles like so. Okay. Does that make sense? Cool. And so the final bit is really just to cut these bits here. And because it's a single-stranded break, it's really easy to fix, right? You just cut it and they're kind of kept in place and you just seal them back up again with DNA ligase. And so, and it's also kind of neat doing this because it really gets at the underlying mechanism of recombination, right? How you generate recombinant chromosomes and different combinations of alleles. Also, it shows really neatly too that during prophase 1 and metaphase 1 of meiosis, these homologous chromosomes cannot come apart because each one is stuck together, right? It's actually technically part of another chromosome via this bit here where you have two different chromosomes making up one chromosome essentially, and the opposite down here, right? So those homologous chromosomes are physically stuck together, right? Not just by an enzyme like cohesin, but literally by sharing parts of themselves. Okay, so we're going to resolve this and we're going to cut. Oops, let's try and write this properly. We're going to cut and shut those two parts of those two holiday junctions, essentially, those two crossing over areas. And what that generates, right, and you can essentially follow it here. Actually, let's put this further along over here. Hang on, just give me a second to think. This is the bit that always confuses me. Yeah, let's stick them there. That's a little bit easier to explain. Then obviously, depending on where that resolution happened, you end up with this recombinant chromosomes like so. Right, so you go from having this process here, fixing the break. We have our exchange DNA here, but the chromosomes are stuck together, and then we end up down here, right, with our different combination of alleles. And that's it, pretty much. Does anybody have questions? Okay, so I'll take a picture of this before I forget and post this up in a little bit. Okay, so what we're going to do now is take a little break. I'm going to look up, see if I can find a recombination video to share with you, see if I can find a decent one. And then from, I don't know, 12:30, let's say, we can have just an open recap question and answer session for anything that you want to go over that will be on exam 2. So again, remember that sex linkage, sex determination mechanisms, non-Mendelian genetics, which is a whole bunch of different stuff, epistasis, codominance, incomplete dominance. I'm sure there's another one in there somewhere, which I've forgotten. Yeah, gene bioenvironment interactions, for example. Then we're going to have linkage, both 2-point and 3-point crosses. And then finally, DNA replication in prokaryotes and eukaryotes, right? So if you have any questions about any of those things, spend the next like 15, 20 minutes or whatever going back through your notes and see what jumps out that you do not understand, because this is the perfect time to ask those questions, right? Because I really do want you to succeed the very best that you can in your exam on Tuesday. Okay, so I'm going to, I'll actually probably be coming back because I'll be using my laptop, but I'm just going to go and get some water and then I'll start looking for a good video. Okay, so see you back here at 12:30. All right. All right, so yeah, couldn't find a good video yet. One was flat out bloody weird. It doesn't match anything that I've learned about homologous recombination. Um, but I will keep looking. Yeah, off of that. After class today, see if I can find one, and I'll send that link in an announcement if I do. All right, so essentially the floor is open to all of you. This is your opportunity to ask me whatever questions you want about the material that we've covered. Here we go. That's what I want. Or I could just read the news. That's also fine. But it wouldn't be as interesting for you because you wouldn't be able to see what I'm reading. So any and all questions? You can ask whatever you like. You can ask them privately on the chat. You can ask them in out loud. So you're all going to get 100% on the exam then. Okay, so okay, I'm just gonna pick on stuff at random. So what does SRY mean in terms of sex determination without looking at your notes? I guess you can look at your notes, but and what are the two things that make a human male male? Well, I guess three things. Okay, shout 'em out. Male prominence. No, those aren't. Y chromosome is the obvious one, right? What is it about the Y chromosome? What's the big deal? Oh, because it's tiny. It is really, really small. Almost have an inferiority complex about the size of my Y chromosomes. What's the big deal about the Y chromosome? There's very little on there. Sperm producing genes and a few other things. So how does a Y chromosome make me male, essentially? Two big things. Yeah, but what proteins? Or what things? What's the main hormone that people believe makes men men? Men. And women. Yeah, testosterone. Okay, so what does testosterone do? Other than make your voice very deep. Actually, women produce testosterone as well. It's calling it a sex hormone is a bit of a misnomer. No. No, those are other proteins on the Y chromosome. What other genes on the Y chromosome help sperm production? Where is sperm produced? Right. So you have to produce testicles, right, first. And so testosterone, which is produced by the SRY, which is a sex determination region Y, I think, produces testosterone. Testosterone causes, because we have both set or undifferentiated gonads, right, as embryos. They could either be ovaries, fallopian tubes, or testes. Actually, if you look at the structure, they are very similar in structure, right? It's just female ones go up, male ones dangle down. That's kind of main difference, right? So we could go from one to the other. Testosterone pushes it over to being testicles instead of ovaries, right? So that's one stretch, but testicles alone don't make male, right? Because we would still have uterus, vagina, et cetera, or undifferentiated sex organs. So what's the second deal? The name has a little bit, I don't know if you follow politics. The Russia investigation was led by a guy called who? What was his name? Yeah, Muller. So presumably someone also called Muller discovered this factor. It's called the Mullerian degeneration inhibiting or degenerating hormone. Can't remember which. Right. So it causes inhibiting. Thank you. Causes the female sex organs that were present, or whatever, to degenerate. So those fallopian tubes, I don't know what the Mullerian ducts are. I can't quite remember what they're involved in. A and P is not my strength. So it basically, the Y chromosome causes testicles to be made. Testicles cause the female reproductive systems to degenerate and to not form, right? And so those two things together, and I don't think the Mullerian inhibiting substance or hormone is actually produced on the Y chromosome. I don't think it is, not 100% sure. But anyway, that then produces male. And essentially, female is in part, again, it's more complicated, due to lower testosterone. So those gonads aren't differentiated into testes, they differentiate into ovaries instead, and those produce their own hormones such as estrogen and progesterone, which direct development in other ways. So for the ladies amongst us, in many senses, being female is the default option, which is kind of cool. Okay. What else? Let's have a little look through. So I'm just scrolling through. Ba-dum, ba-dum, bum. Another question. How do Drosophila determine sex? Oh yeah, there you go. So I'm going to ask you questions. So SXL stands for sex lethal, but so sex lethal is the switch, right? It's the kind of fulcrum essentially. No, it's not XXXO. That is other insects and nematodes. So the O stands for absence of an X chromosome. So, oops. Fruit flies are XXXY like us, but they don't determine sex the same way that we do. There you go. One too many X's though, Gabrielle. Okay, so how do they do it? We do it with, no worries. Yeah, so it evolved independently. It's called, but ends up with kind of the same outcome in a way, and so that's in a sense convergent evolution, right? They ended up with the same XXXY, but got there through a different route. So yeah, sex lethal is active in females and represses male development, but how is sex lethal controlled? What's the main deal in Drosophila sex determination? Yeah, yeah, no. It's the ratio. So AA, oops, XX, what are you doing? Oh, my cat is insisting on playing with me for some reason. Equals female. Why does a 1-to-1 ratio equals female? Yeah. They don't get the Y chromosome. So, I can't remember what it's called. I think it's called Deadpan. I'd have to check that. But if you have 2AA to X, right? And Y is there, but Y is not important in terms of sex determination. It is in humans and mammals. It's not in fruit flies and insects, right? Right, so it's a ratio deal, right? So 1 to 1, A to X equals female. And 1 to, oops, 5, A to X equals male. And basically there's something on autosomes. I think it's Deadpan. There's two other genes. I can't remember both of their names. But there's essentially something on the autosome which suppresses SXL, sex lethal, and there's something on the X chromosome that activates sex lethal. So you have this kind of balance essentially. If you have two X chromosomes, essentially you have lots of that something that activates SXL, more than enough to prevent the inhibitor doing its job. So you're female. If you have only one X chromosome, you have a little bit of that SXL activator and the same amount of the inhibitor, so it inhibits SXL, you get males. So it's essentially the ratio between the amounts of those two different things with SXL in the middle, right? The more X chromosomes you have, the more of that you have, and that's enough to activate it and overpower what's coming from the autosomes. If you only have one X chromosome, you don't have enough, and this inhibitor rules the day and wins, and it inhibits SXL, which equals males. That help? Okay, so that's why it's a ratio deal. It's not the presence of the Y chromosome, because the Y chromosome doesn't do anything. It too has genes for sperm production and stuff like that, but other than that, it plays no role in anything to do with sex. It's essentially the presence of a Y chromosome equals the absence of a second X chromosome. That's why it matters. Okay, what else we got? Dumpy dumpy dum. All right. If a trait is sex linked, would you expect to see more of it in females, offspring, the same amount in males and females, or more in males? Let's see what other people think. And let's human males and females. And this trait being is recessive. Okay, so you see more in males. Why do you see more in males? That's the next question. It's not good enough to just guess because you could like, I don't know, 1 in 3 chance of getting it right. Why would that trait be expressed, the recessive trait be expressed more commonly in males than in females? Okay. Yeah, both of those are right. So males are hemizygous for genes on the X chromosome. Yeah, it is smaller and has fewer genes, but kind of as with sex determination flies, that doesn't actually matter a great deal. What really matters is that you only have one X. And as Peyton wrote, if you only have, oops, get off. And essentially, it's kind of what she said. If you don't have another copy of that gene to potentially dominate or cover up that recessive allele. If you have a recessive allele for a gene on X, that trait will be expressed. You will see that recessive phenotype. So a good example is hemophilia. So let's just so if you have, let's put that over there so I don't knock it over. Let's see. Shit, is it males that are squares? I think so. Right, so this is pretty sure that's the right way round. And so essentially this individual is XY. This individual probably best written like that, right? So if we use capital H as the dominant allele, so normal, and lowercase h as the recessive allele, right, which is the one that causes hemophilia, this individual does not have hemophilia. This individual is a carrier. And so if we say they're particularly fecund, right, and have lots of kids, right, if we have a female, that's going to get one or other of these X chromosomes and an X chromosome from the father. Maybe just by chance, they got the recessive chromosome, right? So this individual is a carrier. Actually, let's make some more space. If you're a male, however, you get your chromosome from your father is always going to be a Y, otherwise you would be female. And then you have the same probability of getting the other chromosome, right? So you could get the dominant allele, or you can get the recessive allele. Now, because you have the recessive allele, oh, craps. Yeah, sorry. Yeah, that would be totally confusing. Yeah, thanks. So essentially, you've got females. It's in red. Right? You're going to get always a dominant chromosome from your father, right? Because if you got a Y chromosome, you wouldn't be female. You would be male, right? And you have a 50-50 chance of getting a recessive allele from your mother, right? So you could either get the dominant allele, or you could get the recessive allele. So you're either going to be normal, or you're going to be a carrier, right? Heterozygous, but you do not express the disease because you have a dominant allele. Does that make sense? Hopefully it does. Now you have the same deal on the other side, right? So now, let's kind of cover that up. Now you have the Y chromosome is from the father, and you have the same 50-50 chance of getting a dominant versus a recessive trait, or allele, sorry, from the mother, right? You could either get this one, or you could get this one, 50-50, right? Because it's random which one you get based on law of segregation, right? So now you could have the dominant allele, in which case you're normal, or you could get the recessive allele. But because there's no other X, if you have a recessive allele for that gene, you will express the phenotype, and yous will be deads. In the olden times, anyway. So essentially, the genotype is kind of the same in a way, right? This one has only dominant alleles. This one has one recessive allele. This one has only dominant alleles. This one has one recessive allele, right? But the phenotypes are different. Normal, 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 recessive trait. And so you will very, very rarely see a female with hemophilia, because it would have to have come from a male who had hemophilia and a female who is a carrier. That's the only way you're going to get two homozygous recessive for that allele. And so the classic sign of sex linkage or sex-linked inheritance is you see far more individuals that are male expressing the recessive phenotype, then you do female. And if you look at any of those lineages for X-linked inheritance or sex-linked inheritance, you will see that it's predominantly a male deal. Like male pattern baldness is another classic example, right? You will very rarely see females with male pattern baldness or just going bald. I mean, there are women that do, but it's far, far more common to see that in males. Color blindness is another one. Or what is it? Green red? Yeah, green red color blindness is also predominantly a male trait, not a female trait. Make sense? All right. Doop, doop, doop. I think that's mostly it for that. Okay, so next. What do I mean by variable expressivity? Yeah, I do always need more coffee. Thing is, this is hard on you lot. Studying for what, 2.5 hours straight it's also kind of tough on the other end too. Okay, what does variable expressivity mean? Oh, you got biostatistics today? Damn. Good luck. I do not envy you one little bit. You should become a biostatistician. They are in very, very high demand because most biologists don't like statistics. Okay, variable expressivity. Not in different organisms. No, definitely not in different organisms, but different individuals. Right, so is that what the slide says? Ah, that's a poorly written PowerPoint slide. Yes and yes. Basically, phenotype or severity of phenotype. So that's why individuals would be better because organisms can actually mean like different species and stuff, which is uh, not very useful because one would expect it to be different between different species. Okay, so God, my typing sucks. Okay, so this isn't variable expressivity because variable expressivity is you're expressing the phenotype, but you're expressing it to different degrees. How would you describe if you have two individuals, they both have the same genotype and one has the disease and the other one does not? What would you call that? It's on the same slide. Yay! That's right, incomplete penetrance. And so that basically means that you look at a population that are exactly the same genetically, and you have less than 100% expressing the same phenotype. So essentially that phenotype has not penetrated to all of the population, right? It's only made some of the way through for some reason. So a good example of variable expressivity is something called mitochondrial disease. Incomplete penetrance is a little bit harder to think up examples for. Yeah, well, actually, it can vary from being very mild, you're just like a little bit tired and stuff like that, to being lethal. And it can potentially be the same genotype causing that whole spectrum. And so it's basically due to mutations in mitochondrial DNA. But because you have more than one mitochondrion per cell, not all of those mitochondria are going to have the same copies of that mutation, right? So even though you can essentially be, I don't know what you call it, heterozygous, that's not really quite right with mitochondria. It depends on the ratio of essentially disease mitochondria to non-disease mitochondria, and where that ratio exists in your body as to what phenotype you express and how bad it is. For example, if you've got like a high mutant ratio in your muscles, then you'll get tired extremely easily after doing exercise because you won't be able to make more ATP. If you have it in your brain, then your muscles might be fine, but you might be mentally retarded. So it varies hugely. Actually, mitochondrial disease was the impetus for three-parent babies, if you've ever heard of that. There aren't very many of them around. So essentially you have, is that in the film? Don't think I've seen that. Two nuclear DNA parents. So, which is standard, right? We're all like that. And one different mitochondrial DNA. Yeah. And so the thing that actually the law was changed in the UK in response to because it wasn't allowed to do that, because this is a pretty extreme form of IVF essentially, and it was from a woman who had because mitochondrial DNA is from the mother only, right? It doesn't come from the father, and she had, I don't know, 6 or 7 children, and every single one of them died before the ages of, I don't know, 4 or 5, something like that, which is quite horrible. Yeah, that's right. So essentially, you fertilize a zygote, right? So you have the two parents' genomic DNA together. Then you take the nucleus out of that, and you have another zygote which was fertilized by, I don't know, whoever else. Take the nucleus out of that and put the other one into the now empty cell. Yeah. Because that's the only way that this woman would be able to have children that wouldn't die, right? So it's pretty wild. Anyway, so mitochondrial disease is a good example of that. What else? What else? Let's jump around a little bit. Okay, what's the role of single-stranded binding proteins in DNA replication? Because there's going to be a bunch of questions like this in the exam. Just basic recall. That's correct. What's okay? What problem does? Hopefully, I'm not giving you exam questions here, but maybe I am. I can't remember. Yeah, I get to eat all of it. Woo! Okay, so this is another. So the question that I just asked is uh, essentially I'm asking the same kind of question that I did before, but I'm asking you the opposite. So I'm not asking you what does DNA gyrase do, but what problem does it solve? Not so much from recoiling. There's another word goes with coiling just in front. Yeah, supercoiling. How does it do that? Correct. It cuts the DNA, lets the DNA rotate, sticks it back together again. Good stuff. Okay, let's have another little look through. Doo, doo, doo. All right, after DNA has been separated and helicase and single-stranded binding proteins, uh, keep things open. Helicase starts unwinding, makes the replication bubble. What's the first thing that has to happen next? Nope. I mean, yeah, I mean it is like the next phase, but what's the next thing that has to be done? Not polymerase one, not yet. Or yeah, polymerase three, sorry. I'll give you a clue. Yes, exactly. So that's all right. If it's an RNA primer, what's the enzyme called? Give you a hint, just add ASE on the end. Primase, right. Awesome. Candy for you. Okay, let's have a little wander around. Dumpty dumpty dum. So, in many ways, this is actually what I do when I write exams. I've got certain things which I need to hit, and then I'm like, crap, I've got another 10 questions I need to write. And so I'll just wander around through these PowerPoints and go, ooh, haven't asked a question about that yet. And that will be another question. Okay. Here's another question. That just made me think of another one. Okay, so this is a inherent mechanism in DNA polymerases that allows them to fix mistakes they make. All right. What does the exonuclease do? This is like asking questions by text message. Okay, cuts off the wrong base. Ah, excises. Yes. Just remember that's not the same as exercises. Okay, then what? Yeah, fat fingers don't work so well. It certainly does reduce the number of errors quite a lot. Yeah, I know. It's like I'm kind of have like tarantulas wandering over whenever I'm typing. I get that a lot. So the answer is it just keeps on going, right? Keeps on synthesizing. Yeah, it's because the camera is down here. It's at the bottom left of my screen because I have a really thin bezel around my screen. And so there's not actually space to put it further up. So you get like this really weird view of like up my nostrils and my hands and really like large. It's very strange. Okay, another question. Let's have a look through, see if there's anything else. Obviously, we're not going to be able to cover everything in just like 45 minutes or an hour, but hopefully. Oh, you know what? I'm going to put that answer in the exam now. I usually put aliens, but magic works as well. I mean, that's how iPhones work, right? Or at least they used to when Steve Jobs was around. Oh, no worries. You go do that. I'll be posting this up anyway, so you're not going to miss anything. So essentially, when DNA is made, how can you tell which is the correct strand and which is the incorrect strand, right? Because presumably the incorrect strand is the one on which the error has been introduced. So what's the difference between the old template strand and the new daughter strand? Well, that's already been done. We've finished removing all the RNA and that processor is already finished. Nucleotide order should be the same other than for that one mistake. So how can you tell which strand is which? Because you have to pick the one where the mistake is on, right? Otherwise, you'll end up removing the correct base and putting the wrong one in. How do you determine between the two? Anybody got any ideas? No? Good idea, though. I mean, that's how the process is activated, right? So you get like a bulge sticking out. So that starts the mismatch repair process. But the question is, how do you know which one? Is this the right one to keep, or is this the right one to keep? Damn it, you did it for... Oh, here we go. Yes, but which one is not methylated? Yeah, correct. For a short period of time, right? So there's a window of time in which mismatch repair can work because those methylation enzymes are coming along behind the DNA replication process. So if you leave it too long, essentially the parent and the daughter strands will look identical in terms of methylation. And so once that's happened, you cannot fix accurately mismatches in DNA replication. It's going to be incorporated into the next generation. Okay, so obviously that isn't everything in terms of stuff to talk about, but I think that did a decent job of kind of showing you some of the things you need to know and the kind of details. And I can tell you straight up, you will get linkage problems. Maybe one, maybe two, I don't know, maybe three. But it's a big chunk of what we've been studying, so it will be on the exam. And you'll be expected to work out stuff using the principles of linkage. Okay, so make sure you know how to do that. Hopefully not. If you're really good at them, it'll take no time at all, Sky. Um, how much time is the exam? It will be an hour and 15 minutes as before, unless you have accommodations with the DSS, in which case you'll get whatever your accommodations are for exams. So it's an hour and 15. There won't be, again, it's going to be a balance, right? So remember last time you had the chi-square analysis to do, at least one. Oh, I need to get up and stretch my legs. But you also have a bunch of short, simple, short answer questions too. So it'll be balanced, right? You're not going to have so much stuff to do that you will not be able to complete it in time. At least that's the intent. Does anybody got questions? All right. Well, my stomach is telling me that it's time to eat lunch. So if you do have questions about anything in particular, send me an email. And also, we will have time after class on Tuesday if you have any questions you wish to add then. Because obviously the exam you can take it at any point up to midnight on Tuesday. So you'll have another opportunity to ask these questions then. So make sure you study and study hard between now and Tuesday. And that should hopefully pop up any questions that didn't arise today or ones that you don't feel as confident with as you'd want to. All right, ladies and gents, see you at 2 o'clock for lab. which shouldn't take too long today, hopefully, but we'll see you then. Let's stop this.